Hello everyone. I will present you the work that we did with Robin Trombetta and Carole Lartisien at Creatis in Lyon, France. Okay, so the scientific context is the one of unsupervised anomaly detection. In anomaly detection, we try to learn the normality with healthy samples only, and then at inference, try to detect samples that deviate from the normality. This will be our anomalies. In neuroimaging, for instance, this can be lesion in the brain. The proposed method is done in 3 steps. In the first step, we will train an autoencoder to reconstruct patches, healthy patches from the brain. We then hope that we will have a compressed representation, a latent representation here that is meaningful for the healthy patches. We train this autoencoder on every control and with patches samples from every possible location in the brain. In the second step, for each patient, we will sample a subset of all possible patches, run it through the frozen encoder from before, and then have this latent representation. With this latent representation, we will tune a one-class support vector machine algorithm to estimate the boundary, the support of the probability distribution of these samples. Because it is a patient, there can be some anomalies in the latent representation, but the OCSVM algorithm is robust to that if there is only a small percentage of anomalies in the whole volume, and this percentage is a parameter in the OCSVM algorithm that we can fix. Then, once the encoder is learned in the first step, the boundary for each patient is learned on the second step, and then in the third step, we can sample every possible patches in the brain of said patient, run it through the encoder, and then compute its distance to the boundary to get an anomaly score for each voxel that will turn into an anomaly score map for the whole brain. The main novelty here of this method is the tuning of the OCSVM, which is done for every patient, so we hope that we will get some boundary here in the latent space that has some patient-specific characteristics. The training on the control set is done on an in-house T1 and FLAIR MRI dataset, and the testing is done on an open database on the WMH challenge dataset, which comprise also T1 and FLAIR, but from 3 different hospitals. We obtain some quantitative performances with our method that is here and also here, so the 2 columns on the right, and we compare these performances to 3 methods from the literature, 1 which was previously developed in the lab, and 2 of which were the state-of-the-art on unsupervised anomaly detection on this WMH dataset. We compare all the methods on the fair ground, so with no pre-training. Here are some of the visualization we obtained. So this is anomaly score maps for each of the methods. So not thresholded anomaly score maps. We can see that all the methods have pretty different characteristics, pretty different pattern, and we discuss some of that in the paper. Thanks for listening, and don't hesitate to chat with us, to send us a mail if you want to learn more of this, to read the paper, and also to chat with us virtually at the conference. Thanks for listening.